Okay, we're starting to record. All right. Hello, everyone. I'm Jackie Drew, and I'm the president of the Oncology, Hospice and Palliative Care, and HIV/AIDS network within the World Congress for Physical Therapy. And we're very pleased to have Liisa Laakso presenting to us on photobiomodulation therapy for lymphedema. And this is a presentation that she recently did in Cape Town and at the World Congress for Physical Therapy. So this presentation is recorded if people want to look at it again or see it for the first time, and it'll be up on the website for the WCPT IPT-HOPE. So Liisa, I'm going to turn my camera off, and I'm going to hand the hosting over to you.
Thank you, Jackie, and hello to everyone from Australia's East Coast. I'm really pleased to be joining in with you this morning, my time, Friday. I'm really happy to be presenting this. It is a little bit of an extension on what I did in Cape Town. I wanted to just do a little bit more around the basic physics of photobiomodulation therapy, or some people call it laser therapy, or low-level laser therapy, low-intensity light therapy. There's a number of names for it, but the official name, the official mesh term now is photobiomodulation, so that it encompasses laser, LED, and all other forms of light therapy. So that's the overview. I won't waste time on that because we are starting a little bit late, and I'm not going to waste a lot of time on the basic physics. But just to bring everyone up to speed, particularly those people who may not know much about the topic. So laser is an acronym for light amplification by stimulated emission of radiation. And as I mentioned, there are a number of names for that, but the one in red there, photobiomodulation, is the physiological effect, if you like. And then if you're using PBM, I will call it PBM. If you're actually using it as a therapy, then we say PBM therapy. Laser is a specific form of light, and it is propagated in a very specific way, quite different to other forms of light. And there's a lot of literature on this. I won't go into all of the ways in which it is propagated, essentially to say that the light can come in many different wavelengths, and there's a table there of some of the different forms of laser specifically, and the different kinds of wavelengths. The lasing medium is the thing that determines the wavelength of the laser, and you'll see there that most of those waveforms are in a range that those of you who know anything about light would know that most of those wavelengths are in a range that are not necessarily applicable for what we do in physiotherapy or physical therapy. The main ones there are the helium, neon, green and red wavelengths, the ruby, which is a classical laser. But we now see most of the therapy lasers, most of the PBM devices are generated using diodes, and are much more cheap to manufacture than a classical laser device. And so we see lots of different kinds of diodes available on the market, and therefore lots of different price ranges for these devices. The specific thing that makes laser different to other kinds of light is that it has these three unique properties: coherence, collimation, and monochromaticity, or the wavelength being of one colour or the same colour. So on the right-hand side there, you can see the little light globe. That's meant to be demonstrating that the ambient light around us is made up of a mixture of different wavelengths, whereas true laser is just one wavelength of light. So if we look at that in terms of what we know is familiar to all of us, and that's the visible spectrum of light. And typically we know that as a rainbow. If we think about light in that context for a moment, and if we turn our rainbow onto its edge and straighten it out, then one of those strips of colour is a wavelength of light. So down the bottom there on the right, we've got a little red laser pointer. And then if we superimpose that on the electromagnetic spectrum, then we have our visible range of wavelengths of light. And just next to that, to the left, for a short period, a short distance, is the near infrared range, and that's where we have invisible wavelengths of light. So the therapy wavelengths of light tend to be in that near infrared and that visible range of wavelengths. And then, of course, if you are familiar with light therapy in any way, shape or form, physical therapists and physiotherapists have been using ultraviolet light for a long time. So that's on the right there in that image. So if we pull that together in terms of what we know about different wavelengths and the EM spectrum, then you can see I've just put an example of some of the wavelengths of light that are in use today for different reasons. CO2 is used in surgical applications. Neodymium YAG on the right in the green is also used in surgical applications. The He-Ne, the helium neon, and the gallium aluminium arsenide, so the 820 nanometers is in the infrared range, and the 640 nanometers is in the visible range. It's a very tight bandwidth of wavelengths in which therapy lasers or therapy PBM devices actually sit. So this has relevance in terms of the depth of penetration of that light. The visible wavelengths of light in that range between 600 and 750 nanometers don't penetrate very far. They are absorbed very well superficially and are really good for treating wounds and superficial lesions. And by contrast, on the right, that image is just an indicator. It's not an actual representation of what happens, but it gives you an indication that the invisible or infrared wavelengths of light bypass the superficial layers of tissue and can penetrate deeper. And generally speaking, they're used more frequently for musculoskeletal applications. Jackie, I can see somebody's name in the top right-hand corner. Can I just digress for a moment and ask, does that mean that people can ask you questions along the way, or should I just keep going?
It's really up to you. Sometimes people will ask a question in a chat, but if you'd prefer to go through the presentation and then answer questions at the end, that's what people typically do. So if that's okay.
Okay, I'm happy to do that.
All right.
Thank you. Okay, so moving on then, I mentioned about diode lasers. They can be quite expensive, but they can also be quite cheap to manufacture and to purchase. In physical therapy, physiotherapy, we see lasers that can be of a single diode there. So in the middle image there, that's a device that has just one diode in it. Similarly with the image down the bottom. And in the image on the top left, we've got a single diode PBM device in the middle of that little array of diode probes, and the two on either side are cluster heads or shower arrays or multiple diode devices. So they may have diodes that are of just one wavelength or multiple wavelengths, so that you get different depths of penetration from those different wavelengths of diodes that are represented in that cluster head. So there's lots of different ways in which PBM can actually be applied to an individual. And that also includes scanning lasers, which are fairly typically used for larger areas of the body, and most typically in applications for lymphedema. And that's why I have presented some of the basic physics around the need to understand that you can apply it using a single diode to really target an area specifically, or you can apply it using a scanning laser to cover a large area, which is often how we see lymphedema being presenting in a clinical setting. The dose calculation factors do come into it when you're using a scanning laser. It can be difficult to replicate your treatment and to be really sure about exactly what kind of dose you're using once you start to utilize a scanning laser. It is possible with some of the newer devices now to have a better understanding of what dose you're applying, but it still has its challenges for that purpose. Okay, so moving on to topic 2 then, the basic cellular mechanism of PBM therapy. And this is just a single slide. I'm not going to go into a great deal of physiological discussion around this. The primary mechanism is the absorption of photons by cytochrome C oxidase within the body's cellular powerhouse, which is the mitochondria. This has been shown time and again in all kinds of research in vitro and in animals. So we do know that, as you can see there on the bottom right, that different wavelengths of light will be absorbed preferentially in the body, and some wavelengths are better absorbed than others. So that becomes critical when we're thinking about using PBM for lymphedema. So moving then on to the specifics of using PBM therapy for lymphedema, if we start to think about the physiological effects, it was in the early 90s when colleagues of mine in South Australia, Anne Thelander and Neil Pillar, were starting to use laser as a method of treating their patients with laser therapy, because they were hypothesizing, based on some of the earlier literature, that it might be able to stimulate lymph vessel regeneration and increase the diameter of the lymphatics. And from there, they hypothesized that it might actually increase the contractility of the lymphatics and result in removal of the accumulated proteins and fluids that typify lymphedema. So they started to do a little bit of investigation around this from a clinical research perspective. Around about and informing their hypotheses was some of the very earliest research, and some of that early research has implications for how we think about PBM therapy and its impact on tumours specifically, on metastases and on lymphedema directly. So if we look at the chronological development of our understanding of PBM therapy and lymphedema, way back in the 80s, Chris Castell and others were looking at the effect of light therapy on immune modulation and found that it was possible to redirect edema, and they hypothesized via the sodium ion channels. Levens and their group looked at this specifically in animals and found that, in fact, the lymphatic vessels did dilate in response to PBM, and I'll show you a couple of slides about that in a moment. A little bit further on in the early 90s, Honmura and colleagues found that PBM increased the microvasculature and increased vascular permeability in certain applications. Moving further along in the chronology, others found that PBM stimulated angiogenesis, stimulated an increase in blood flow, and from a submolecular or molecular level around cytokines research and the like, others found that NOS and VEGF was increased. So those 2 points there in red are very specific to our understanding of how PBM may have an effect on lymphedema, but how it also has some risk potentially in the case of tumours and metastases. So I will come back to that in a little bit. So if I go back to Leven's work, back in 1987, their group was doing some research on mice. They found in red there, as you can see, that laser, specifically laser, restored lymphatic function faster in the incised vessels of mice compared to the control group. So when I say significantly faster, that restoration of function was in a week compared to many weeks. There were no adhesions, soft tissue adhesions in the mice that were treated with laser, whereas there were, 90% of the control group had adhesions in the soft tissues at day 6. A little bit later, Lievens did the work on dilation of lymphatic vessels, and then of course we saw the, I've already been through the other points there. So if we look at that sentence in red, transfer that across to the next slide, and actually have a look at what Lievens' group found. This is a couple of images from their work. You can see in this image that V stands for venules, A stands for arterioles, and L stands for the lymphatic vessels or lymphatic channel there. When they applied the laser directly to that area, to these mice, they observed the dilation of all of those vessels, in particular the venules and the lymphatics. So that was a pretty impressive finding in this animal study, and that's what probably was the biggest trigger for Thelander and Pillar when they started to investigate other means of assisting their patients in a clinical setting around trying to manage the lymphedema. So let's fast forward then and look at what the clinical evidence for PBM therapy is for lymphedema, and I'm going to take a broader view first with a couple of systematic reviews and meta-analyses, and then we might have a look at the specific details of some of this research. Lima et al. A few years ago in their systematic review found that there was a reduction in limb volume in groups subjected or the individuals subjected to laser photobiomodulation when compared with other treatments, but there were no studies that they could find that evaluated the risk of metastasis or relapse, and no studies that compared PBM therapy with the usual method of complex decongestive physical therapy. Smoot subsequently did a systematic review with a meta-analysis, and they also supported that finding of PBM therapy reducing limb volume in the management of breast cancer-related lymphedema. In a little bit more detail, they found that there was only a limited number of studies with small sample sizes, but the pooled analyses did support a statistically significant and clinically meaningful reduction in limb volume across those studies. And that was immediately at the conclusion of treatments that included PBM therapy by direct comparison with those groups that did not receive the photobiomodulation therapy. The other thing that was really interesting in their review was that there was a possibility of pain reduction with the use of PBM, and that is certainly something that we've seen in other literature around PBM therapy, that it is very useful for reducing pain across a whole range of musculoskeletal conditions. So that's not surprising to me at all, but I'm not going to be talking about that at any length today. So if we look a little bit closer at some of the research that was included in those reviews, Karate and colleagues was the first one to do a double-blind placebo-controlled trial, a proper rigorously controlled clinical experiment to understand the role of laser in this case post-mastectomy lymphedema, they referred to it. They used 904 and 104 nanometer wavelength, a fairly low power output there of 5 milliwatts, and they treated the axilla 3 times a week for 3 weeks, and their total energy for each treatment was very low, using that little device there that's in the left-hand corner, so just a single diode laser. They found that 2 cycles of laser was better than 1 cycle, so a cycle being that, if we go back to that first slide, a cycle being 3 days a week for 3 weeks, so that was a treatment cycle. If they repeated that, then they saw improved reduction in limb volume, and decreased bioimpedance and tissue hardness measures. There are some positives and negatives about that particular study, as there always is with research. There's things that you can critique. But essentially, this was one of the first studies to really show in a comparative way that the benefits of laser or PBM. I draw to your attention on the right-hand corner there, only 31% of their participants actually improved. And I asked the question, what about the other 69%? So, whilst there was benefit to about a third of their clients in terms of limb volume and those other objective measures, there was still a significant number of their participants who didn't get a significant improvement. Caviani, a little bit later, did some work on another wavelength of laser, 890, still using a low energy density, and they also used a spot application, extending that over a 4-month period of 18 sessions, and they noted the indirect measure of arm circumference being improved, and therefore limb volume being improved as well. And they note that the matter of a reduction in pain. Small sample size yet again. Lau and Cheng from Hong Kong did another study in Chinese women. And I think it's nice to see some cultural aspects being brought into this. Can we see the same things occurring in populations outside of the usual Anglo-Saxon realm or the Caucasian realm? So they too use some different wavelengths, 808 and 905 nanometers there, but still in that infrared range. They used a slightly higher dosing, 2 joules per square centimetre, a little bit more in numbers, in terms of participant numbers, which is good. And they did 12 sessions over a 4-week period, again to the axilla, not to the whole arm. And they noted a significant reduction in arm volume and a cumulative improvement at follow-up as well. I can't remember how long that follow-up was for, but it was interesting to see that reduction continues beyond the end of treatment. So some interesting clinical findings there. Smoot then tried to bring all of that together in their systematic review to give us some indicators of how we should be using PBM therapy for lymphedema, and they summarised their findings in this way: that you need to have at least 17 minutes of treatment in a session. That's not a good way of describing PBM dosing. You need to have a mixture of ways of describing it, but they do go on to say that the dose of around 1.5 joules is indicative of the dose required. So that dose will change depending on the device that you're using and its power output. They landed on 904 nanometer laser being the best wavelength, probably partly due to Lieben's original work, but also the fact that most people treating lymphedema tend to use 904 nanometers. I'm not convinced yet that you have to restrict yourself to 904. I think it is possible to get benefits from other wavelengths, as we've seen. And then you need to treat at least 3 times a week for up to a month. And most of the research, as we've seen, has been applied to the axillary region to try and open up, dilate those lymphatic vessels to encourage the lymphatic flow. And then, of course, based on Karate's work, it's probably better to follow up with a second cycle of PBM at a later date if you can get your patient back, or if they're interested in doing so, to accentuate the findings or to build on that original outcome. So I'm going to have a quick look at safety now. This is obviously something that those working in the field will be concerned about with electrophysical agents. Are they safe for use in cancer? I'm going to restrict my comments to PBM therapy. I think at some point you'll have a speaker talking about safety in general of electrophysical agents. But before we do that, we need to have a quick look at cancer biology. It does have reference and relevance to the use of electrophysical agents for lymphedema. We know that metastases can be activated or facilitated through hematogenous means, but we also know that metastases occur through lymphatic flow as well. And obviously the lymph vessels themselves can be adversely altered by cancer treatment and can be affected by nitric oxide and other factors within the body, including prostaglandins and anything that affects autonomic regulation. You can see there's a fair bit of information around how cancer biology contributes to metastatic spread. I draw your attention to this second last point about VEGF, and you noted earlier that in one of the studies they found that VEGF is actually increased with laser or PBM therapy. So the question that has always concerned me is if in treating breast cancer related lymphedema we're getting a reduction in limb volume, what are we actually doing to these other factors that may in fact be increasing the possibility of lymphatic spread? So I've done some work in this space. We started off by looking at animals in the first place. We injected malignant melanoma, human malignant melanoma cells into nude mice, and nude mice being immunodeficient. We utilised a really low dose, 1 joule there, and a really high dose, 12 joules, and irradiated 4 spots at each tumour site using a wavelength of 780 nanometers, so still in that infrared range. We treated those mice 3 to 4 times a week for 4 weeks, and we found that there was no effect at either dose, either the high or the low dose, on tumour burden. So that was encouraging to us. We then went backwards in terms of our translational research. We had a look in vitro in cell cultures, examining the possibility that different wavelengths might have an effect on human breast cancer cell lines, and we tested human glandular mammary adenocarcinoma cell lines as well as human ductal mammary carcinoma cell lines, and we utilised a range of doses and wavelengths to have a look at what the relationship might be. Now, for the ductal carcinoma cell lines, there was really minimal effects at all the wavelengths and doses that we tested, so that was a good thing. For the glandular adenocarcinoma cells, with one exposure to these cells in a culture dish, okay, so we're talking about benchtop research here, and you can see with the red dotted or dashed line there that as dose increased, so did the potential for these cells to proliferate. When we exposed the cells to 2 treatments of the laser, you can see that red dashed line normalizes to the baseline, and then when we did 3 exposures, we actually saw a reduction in proliferation or a lessening in the cell numbers. So that was really an eye-opener for me. We saw a similar thing for 904 nanometers. This is the typical wavelength that is used for lymphedema treatment in a clinical setting. So the red dashed line there with 1 exposure increases with increasing dosage. It normalizes with 2 exposures, and the red dashed line reduces or decreases from a linear regression perspective as dose increases with the 3rd exposure to the laser. Really exciting stuff. So from our perspective, when we looked at this data, obviously different cell lines show different effects when exposed to different wavelengths of PBM. Repeated exposure using some wavelengths may be safe or may indeed have a protectant effect if we were to transfer that into a clinical model, and that PBM does appear to be safe for use with ductal carcinomas. Getting there, folks, almost finished. So at point 7 then, the effects of PBM on cancer cells and the implications for clinical use. Our work has been extended and built on by others. There is other work in this field as well. Samoilova and colleagues, she found that in general the in vitro results suggest an effect at the systemic level when you're using both visible and infrared light, that those wavelengths may stimulate the growth of human skin cells, but they simultaneously downregulate the proliferation of tumour cells, including breast cancer cells, and those results argue in favour of the oncological safety of PBM therapy for breast cancer patients post-surgically. In some work done by Mikhailov in Russia that began in the 90s and has had 10-year follow-up now, this is a case series, so we need to be mindful of that. There are always issues with that kind of research, but Mikhailov has been using laser for a long time to treat various problems with patients post-operatively after breast cancer, and has looked at the recurrence rates in the group of patients that he's applied laser to, and these results examine 41 patients who've received laser at various wavelengths and doses, and they've looked at disease recurrence, and you can see there the data about disease recurrence at 10-year follow-up, pretty impressive, and the figures compare really well with known survival data for patients generally who've had breast cancer. So, you know, there doesn't seem to be any indications there around safety issues for using PBM therapy for lymphedema. So my last slide then, I want to leave a message with you that electrophysical agents are a valuable adjuvant to your total management plan for lymphedema. And that should include, of course, your complex decongestive therapy, your massage, your compression, your exercise and self-care. The way that you use the laser might be for small areas of induration of fibrotic tissue. It could be in the axilla. Colleagues who work in this field also use it, use scanning laser to treat the whole of the limb as well. So there are various ways that you can use the laser or the PBM for lymphedema. The overriding thing is to hopefully treat the signs and symptoms of lymphedema early, those signs and symptoms that may prevent your patient from moving adequately, engaging in physical activity and exercise that is so important for self-care and survival in a cancer setting. And of course, by treating and controlling the signs and symptoms, you're assisting the patient to move towards self-efficacy, which is such an important thing when we're seeing patients in this kind of field. So thank you. I hope I haven't taken too long, and I've caught up a little bit of time for you, given that we started a little bit late.
Liisa, this was just an excellent presentation. I want to thank you for the research that you've done in this area to show the safety issues with this. And what I'd like to do now is open up the floor, the online session. If anybody has any questions to ask Liisa or comments that they'd like to contribute, you can either use your microphone or the chat line. Are there any questions?
People would like to go home by the sound of it.
Thank you so much, Liisa, for talking with us today. Can you hear me okay?
Yes, I can.
I'm so happy to have, this is extremely fascinating to me. And so basically the bottom line is that you've shared with us today is there are, in a way, cumulative positive effects of the laser therapy, at least from the research that you've done and others have done. Is that the take-home message?
That would be the take-home message. I think we've been using it in Australia for a long time. I think Australian physiotherapists have been frustrated by the problems that exist around treating such an such an intractable and incurable condition that that they've reached out to try a number of methods. Perhaps in the early days, we weren't thinking necessarily about safety so much. We were willing to try anything to help our patients, but the patients themselves now in Australia have been advocating for the use of laser therapy amongst their patient groups and support groups. The women with breast cancer around the world, I think, are terrific in supporting each other, and the word spreads via those types of groups. And we see women actually seeking out laser therapy now because it has such a positive effect in those individuals who suffer from this condition.
Can I ask you then a follow-up question? When is it efficacious to start the laser therapy? So when in the process, how early, I guess is what I'm asking.
Yeah, look, we've done, thanks, that's a really good question. We've done some clinical audits, some chart audits of clinicians who use laser as an adjuvant or an adjunct to their normal treatment, and they find that, generally speaking, if their normal treatment is not producing the benefits they would like to see, then they add the laser in. What I'm basically saying with that response is that you can start at any time if you think that it's going to be helpful and potentiate your the effects that you're getting from standard treatment. The other point when a lot of the clinicians here who use it find that it's very useful is when you get those little patches. I'm not a lymphedema therapist at all, so my description of this is probably not particularly accurate, but I'll try to explain it. When you're treating someone with long-standing lymphedema, you get patches of fibrotic tissue, of hard tissue, and that can, in fact, block the flow of the lymphatic vessels distally. And so colleagues who treat these women with these kinds of patches of hardened tissue say that they use the laser on those areas specifically to try and soften those areas, and they see that it's of great benefit for those long-standing lymphedematous arms. So it does soften tissue, but it also promotes lymphatic flow.
So this could be done anytime as warranted or as determined by the physiotherapist, and even during and when chemo and radiation therapy is going on?
Absolutely, and I think there's some really good data now from across the world that indicates that radiation dermatitis can be prevented if you apply the laser during radiation. So I think it's something also to consider in that particular setting. We've done a few single cases looking at this. Most recently, I had a colleague in Melbourne who was seeing a patient that had previously had radiotherapy, who had had a really bad reaction to the radiotherapy along her scar line, along the mastectomy scar line, and they needed to retreat that area with radiotherapy. So there was a great deal of concern about the scar breaking down and a really bad radiation dermatitis reaction. So my colleague convinced the treating oncologist that it would be worthwhile trying the laser. The patient was very happy. She was in fact the one who came to see my colleague about what could be done to mitigate the risks in this setting. And they started to treat it at reasonably low doses along the area of the scar before radiation commenced and through the radiation period. And they had a really nice effect from that. The area of breast tissue that was treated with the laser looked very healthy, whereas other parts of the radiation field did start to develop the radiation dermatitis. So it was a nice little single case. I wish I'd taken some photographs to demonstrate the effect locally.
Were they using special creams when they were immediately following each radiation therapy treatment?
To be honest, I'm not sure.
Okay. Thank you very much, Liisa. Thank you again.
Thanks, Ed. Good to see you.
Good to see you, too. Thanks for getting up early.
Any other questions or comments? You know, I do have one question. The first study that you showed, I think it was by I wrote it down. Karaoke or something. Where only 30. Yes, karate. Where only 31% of the people had an improvement. When I first saw this slide, I thought, well, maybe they were treating different levels. So maybe some people had the hard therapy, tissue, the hardened tissue, compared to more fluid lymphedema. But with what you're saying from some of the later studies, it seems to work well, even better maybe with the hardened therapy, tissue. I'm sorry. So, do you know any reason why this didn't work? Like was it because they were not treating the patients as long as some of the other later studies, or the dosages were not the same?
Look, I think the main thing here is that they were treating only the axilla, so they were probably getting a dilation of the lymph vessels in the axilla and promotion of lymphatic flow proximally, but they weren't irradiating the rest of the limb. So it probably serves as a teaching moment to suggest that we should probably be treating the axilla as well as the remainder of the limb. You'll notice that this particular device is a single diode laser, so it takes a while to treat the number of points in the axilla. I think they were treating 17 points in the axilla. If you treat at a minute per point, that's almost 20 minutes of your treatment time. Whereas with the scanning laser, you can set the scanning laser up. You set your patient up in a nice supported position where they're less likely to move or want to move. You set the scanning laser over the top of the limb, and then set the laser to scan along the limb, and then you can leave that, if you trust your patient, you can leave that and go away and do something else in the meantime, and then come back, and then, if you need to, set the patient up in a slightly different position so that further scanning can occur on the posterior part of the limb if you've done the anterior part, for example, or you can use a handheld laser like this particular device and just treat some specific spots, and that reduces your overall treatment time. So I think there are issues around where we target the treatment that we need to do some more work on to find out what the best protocol should be. Okay.
Wow, this is amazing. I am not lymphedema certified specialist, but there were so many people within in my section, the oncology section in the USA, and I don't know of any of them that are using laser. Do you know of anybody, Ann, that might be using this?
Yes, can you hear me?
Yep.
Yes, I have some colleagues that are using it in the clinic where I sub in, and as far as I know, it's been pretty successful on almost everybody. There was one person they were asking me about recently who had that kind of thickening tissue hardness that was a little more diverse, or rather extensive, and they were having some difficulty with minimizing that in that individual. But overall, they've had very good success. I'm not sure of which laser they're using, and they're using it in combination, of course, with other kinds of treatments, such as wrapping and deep tissue work. and exercise. So,
that's great to hear.
Yeah.
Yeah, I think there's a lot of my friends that are going to be getting a copy of this recording. This is really amazing. I'm so happy that you agreed to present this, and hopefully we gave you a little bit more time, because I know things were short at the World Congress. We, just didn't have a lot of time for the speakers that were there.
Well, I'm really grateful for the bit of extra time, because there was so much there, and there is so much to say. I could probably say more about this as well, but I think we're getting to the point with this field that we can be fairly confident that it's a safe method. But as with everything, we can't be entirely sure, and to do a research study on this would be, there might be some issues with ethics, but I think the data that we have now would allow us to make an argument that we could perhaps extend this to other forms of cancer-related lymphedema or other forms of cancer-related symptomatology, and that's certainly what we've been looking at. I think the main thing is that even though we can see an increase in proliferation of cells in culture in certain circumstances, the fact that when you repeat your treatments, that has a reducing effect on that proliferation. I think that's a really exciting thing, and perhaps using laser or PBM therapy before people start to undergo radiation or before they have surgery for wide local excision or breast surgery, then maybe there's a place also for it to be used as a preventive measure. They're ideas for the future. I think we have to bring the oncologists on board if we're going to be doing those kinds of things, and convince them and get some champions there around this.
Right.
But the only other point I'd like to make here is that if you're ever concerned about this and you're not sure as to whether you should use it or not, then start using it during the time a patient is receiving their treatment, either their chemo or radiotherapy. And in that way, you can be sure then that if you are proliferating any cells, theoretically, if you are proliferating any cancer cells, then the chemo and the radiotherapy will be mopping up those proliferated cells. So maybe start there and get some confidence in using the laser, and then you'll find that as you see the results, that you'll feel more confident to use it at other times during the continuum of a survivor's journey.
That's a good strategy to use. And then, in terms of improving scar management and pain, I mean, you didn't really address that, so we're going to have you come back, Liisa.
Sure, I'd love to do that. I think there's a really good place for that as well.
Yeah, this is very valuable. Well, I just wanted to let you know that our Education Director, Nina Chegbo, she couldn't be here tonight, but she will be sending you an International Scholar Award. It's a bequest to people that come and present their work on our website. And I just want to thank you again. This was really good information, and like I said, I will be sending it to a lot of my colleagues who are working in this area. I think it's going to make a real difference in patient care and in patient outcomes. So thank you again.
A real pleasure.
All right, and I will say good morning to you and good night to Anne.
Okay, and take care, everyone.
Yes, you too. Good luck, Liisa. Thanks again.
Thank you. Bye-bye.
Bye-bye. So I'm going to end the recording, and this will be placed on our World Congress website so that other people can learn from this. Thank you again, and good afternoon, morning, or evening, depending where you're from. Take care.
Okay.
Bye-bye.
Thanks again. Bye-bye.
Yep, bye-bye.